Good afternoon, everyone. I am Dr. Shaheen Sultana, Professor, IIMT College of Pharmacy. Today, I am giving a presentation on Gastroretentive Drug Delivery System. Now, gastroretentive dosage form are oral formulation that are designed to be retained in a stomach for a prolonged time and thereby enable sustained and prolonged input of a drug or drug release to the upper part of the GIT tract. This technology has generated enormous attention over the last few decades due to its potential application to improve the bioavailability of some drugs via prolonging the retention in the upper GIT tract. So it is used for site-specific targeting also, and the drug is showing poor bioavailability because of the absorption problem in the GIT tract. They can be overcome by this gastroretentive dosage form. What is the need for gastroretentive drug delivery system? So the drug having low solubility at high pH, such as furosemide, verapamil, chlordiazepoxide, etc., they all needs gastroretentive drug delivery system, which is having the narrow therapeutic index, narrow absorption window in the GIT tract. So they can use gastroretentive drug delivery system. They can be formulated as drugs that degrade in colonic environment, example metronidazole, captopril, ranitidine, which degrade in the colonic environment of the intestine. So they will be retained in the stomach for the long period of a time. So they will not be allowed to degrade in a colonic environment or in the alkaline environment of the intestine. Drugs that disturb the normal colonic microbes like antibiotic used for the eradication of H. pylori, such as tetracycline, clarithromycin, amoxicillin, etc., they can be given as gastroretentive drug delivery system. Now, the drugs, those acting locally in the stomach, like antacid, drugs for H. pylori, they want the action in the upper tract of the GIT tract. So here the GIT drug delivery system as gastroretentive drug delivery system, it is highly desirable because the action it is desirable in the upper tract of a GIT. So the formulation can be prepared as gastroretentive delivery system. Drugs which is absorbed only from the upper part or the upper part of a GIT tract, such as calcium supplement, they can be prepared as gastroretentive drug delivery system like floating microspheres. Limitations of gastroretentive drug delivery system means the drug that is not feasible for those drugs that have solubility or stability problem, that is unstable in the GIT tract. If you retain the formulation there for a long period of a time, if they can cause irritation, if they are unstable, they degrade. So no need to prepare the gastroretentive drug delivery system for such formulation. The system that requires high level of fluid in stomach, because there are one type of gastroretentive drug delivery system which is floating. So they want high level of a fluid, high level of a water in the stomach. So in that case is also there is limitation for the gastroretentive drug delivery system. This is the disadvantage. For floating, you want high amount of water in the stomach. Otherwise, it will not float and your purpose will be destroyed. The drug that are unstable in the gastric environment, they cannot be suitable for gastroretentive drug delivery system because they can degrade in acidic environment. The dosage form should be administered with a full glass of water if your dosage form it is floating based drug delivery system. Drug which is having irritant effect on a gastric mucosa, they cannot be delivered as gastroretentive drug delivery system. Drugs which absorb throughout the GIT tract and which undergo the first pass metabolism, they are not the desirable candidates for the gastroretentive drug delivery system. Now here is the diagram for the different types of gastroretentive drug delivery system. We can see here that there is modified shape system. In modified shape system, you can prepare different formulations of different forms or different shapes that can retain in the stomach for long period of a time. Hydrodynamic balance systems are there. This system, it is a type of a floating drug delivery system that when they absorb the water, their density will be decreased to 1 or less than 1, and therefore they will be float on the gastric environment. Now, high density system, these are the formulations whose density is more than 1. So when their density is more than 1, they will be settled at the bottom of the stomach, which is near the pylorus. So they withstand all the peristaltic movements of the stomach and remain in the stomach for a long period of a time. Expandable system, that when in contact with the GIT fluid, they will become expand. When they expand, they will retain in the GIT tract or the lower bottom of the stomach and cannot be removed at the site of application or from the site of application. Floating microspheres or floating drug delivery system that have density less than 1, so they will be float in the stomach for long period of a time. Similarly, mucoadhesive drug delivery system that will show adhesion with the tap, that will show adhesion with the mucous membrane because of some mechanism involved and remain in the stomach for long period of a time. Now, when first form, it is in detail, one by one, hydrodynamic balance system. This type of system, they contain drug along with the gel forming hydrocolloid. This system, it is remain floating on the stomach system, that is buoyant on the stomach content. These are single unit dosage form, means they are like a tablet, or they contain one or more gel forming hydrophilic polymers. They usually prepared with the combination of the polymers. The incorporated floating materials enable the device to float for a long period of a time, like more than 12 hours or 24 hours. Such effective drug deliveries, it depend upon the balance of drug loading and the effect of a polymer on its release profile. Now, raft forming system incorporating alginate gels, this system, they have received much attention recently for the deliveries of the antacid and gastrointestinal infections. So there is the mechanism, is the formation of the raft. Raft, it is basically the gel forming layer. A layer of a gel, it will be formed inside the GIT fluids. So mechanism involved in raft forming system is the formation of a viscous cohesive gel in contact with the gastric fluid, wherein each portion of a liquid, it will become swells and it forms a continuous layer which is known as the raft. This raft float on the gastric fluid because of the low bulk density created by the formation of carbon dioxide. Usually the system contains a gel forming agent which is known as alginic acid, carbonate components which is known as sodium bicarbonate, and acid neutralizer which forms a foaming sodium alginate gel or raft in contact with the gastric fluid. Bioadhesive or mucoadhesive system. Now, bioadhesive system are those systems which can bind with the gastric epithelial surface or a mucin, and therefore they will result in increase in the drug absorption process in a site-specific manner. Actually, this type of mucoadhesion, it involves different mechanisms. Like your polymer, if it is positive charge, it can interact with or bind with the negative charge of a cell membrane. Negative charge of a cell membrane, it will bind. Because of this binding, there is an adhesion between the two surfaces. One is formulation and your one is the gastric mucosa. Now this approach involves the use of biomucoadhesive polymer that can adhere with the epithelial surface of the GIT mucosa. The mechanism that involves is the formation of hydrogen or electrostatic binding can be formed with the biological membrane or at the mucous polymer boundary. Now here the diagram, it is shown where the mechanism, it is depicted. Now the mucin chain, you can say here, the mucin chain first interact with the polymer chains, then there's a contact of a polymer chains with the mucin layer, and finally there is interpenetration of a polymer chain with that of the mucus. High density system, these system include high density. They have a density of more than 1 gram per centimeter cube in the stomach content. So when the density is more than 1 gram per centimeter cube in the stomach, then it will be settled at the bottom of the stomach, which is near the pylorus. So this goal is achieved by coating the drug with a heavy inert material such as barium sulfate, zinc oxide, titanium dioxide, iron powder, etc. The heavy pellets, it will remain longer in the stomach since they are positioned in the lower part of the antrum or near the pylorus. Modified shape system. These are non-disintegrating geometric shape or different shapes that you prepared from celastic elastomer or rubber or extruded from polyethylene blends which extend the GIT or gastric transit time depending upon the size and shape and the flexural modulus of a drug delivery device. Now, expandable system. This system, when comes in contact with the gastric fluid, it becomes, take the water, and it becomes swell. After swelling the system, it prevent their passage. When they swell, then their size will become increased. That prevent the passage from the stomach through the pylorus and get retained in the stomach for a long period of a time. It can be cleared from the diagram. You can see here the dosage form. When it comes in contact with the water, it becomes swell. After swelling, its size is increased. So it will be retained in the stomach. So these type of system, they are known as plug type system because they have a tendency to remain locked in the pyloric sphincters. Now the magnetic system, in this system, the dosage form, they contains very small internal magnet. And a magnet, it is placed on the abdomen over the position of the stomach. Some magnetically active compound, it is incorporated in the dosage form to achieve the site-specific drug delivery. So this system, it allowed the site-specific drug delivery based on magnetic effect. Then floating drug delivery system. The floating drug delivery system, they have bulk density lower than that of gastric fluid, and it will remain buoyant in stomach for a long period. While the system is floating on a gastric content, the drug, it is released slowly at the desired rate from the dosage form. After the release of a drug, the dosage form, it is empty from the stomach. While such a low density system, it floats over the gastric content, it increases the gastroretention time and therefore provide the drug released at a desired rate with the reduced fluctuation in plasma drug concentration. Now, this is the flow diagram that shows the classification of a floating drug delivery system. There are two types of floating drug delivery system, effervescent and non-effervescent. So effervescent can be the matrix tablet, gas generating system, volatile liquid containing system, and non-effervescent is alginate beads, multi-particulate system, colloidal gel barrier system, microporous compartment system, and hollow microspheres, or which is also known as microballoons. Now, when we talk about the effervescent system, these are the floating matrix system that is prepared with a swellable polymer such as methocell or chitosan and various effervescent compounds such as tartaric acid, sodium bicarbonate, and citric acid. Now, when this type of dosage form comes in contact with the gastric content, the system liberates the carbon dioxide, which get entrapped in a swollen hydrocolloid, which provides the buoyancy to the dosage form. You can see here in the diagram that effervescent substances like tartaric acid, sodium bicarbonate, and citric acid combine with the drug, and there is semi-permeable membrane. So then water, it will enter through the semi-permeable membrane. It will dissolve the effervescent compounds and this result in a release of carbon dioxide. This release of carbon dioxide, it causes the floating power to the system. Different type of effervescent system. There is volatile liquid containing system. The device is osmotically controlled floating system that contains a hollow deformable unit. It is formulated by incorporating an inflatable chamber that contains liquid like etherocyclopentane that vaporizes at body temperature, inflated in the stomach. There are two chambers in the system. First contains the drug and the second chamber that contains the liquid. The device also contains a plug that is made up of polyethylene or polyvinyl alcohol, which slowly dissolves, causing the inflatable chamber to release gas and collapse after a particular time to permit the spontaneous drug release. Gas generating system, such a system utilizes the effervescent reaction between the effervescent compounds, like as previously discussed, that carbonate or bicarbonate, citric or tartaric acid, the reaction causes the release of carbon dioxide that get entrapped inside the gel hydrocolloid layer of the system and therefore decrease the specific gravity of the formulation and making it to float over the food or over the time. Now, matrix tablet, the single layer matrix tablet, it can be bilayer or it can be triple layer. So tablet can be used to extend the gastro retention of a dosage form. Now, single layer matrix tablet, it is prepared by incorporating bicarbonate in matrix forming hydrocolloid such as HPMC, chitosan, alginate, or other polymer and drug. In bilayer tablet, it is made up of two layers. One contains a quantity of a drug that will provide the immediate release, while the second layer contains a quantity of drug that will provide the extended release for 12 hours or 24 hours. The rapid release layer, it disintegrates immediately after administration and therefore provide the immediate effect. While the matrix layer that remains in contact during the passage of a drug through the GIT tract, it provides a controlled release in order to maintain the plasma drug concentration for a prolonged period of a time. So we can see here third type, which is the triple layer tablet. It contains the inner core layer that is sandwiched between the two surrounding barriers, which is the polymeric layer. We call a drug controlling membrane A and drug controlling membrane B. So here in the, you can see here all three diagrams it is shown. That first shows the single layer tablet, and the second is your bilayer tablet, and the third it is the B, and B it is the form of a triple layer tablet. And you can say here the A form, which is the bilayer tablet. And single matrix tablet, it contains only the single layer. The drug, it is mixed with the polymer. Non-effervescent system, the mechanism here, not the effervescent compounds. Here is some different mechanism involved that causes the floating of a dosage form in the stomach content. So the formulation, which is prepared by mixing the drug and the gel forming hydrocolloid, it swells in contact with the gastric fluid, and after swelling, it attains the bulk density of less than 1. So when the bulk density is less than 1, then the system remains float in the GIT tract or in the fluid of the stomach content, and allow the sustained release of a drug through the gelatinous mass. You can see here in the diagram that the drug, it is mixed with the polymer. Then the system swell after formation of a gastric fluid, and its density become less than 1. And then the system start floats. After the system start floats, the system slowly release the drug for a long period of a time. Now, the various type of non-effervescent floating drug delivery system also, it is the hollow microspheres or the microballoons. It is a type of a non-effervescent floating drug delivery system. You can see here the microballoons or hollow microspheres that is prepared by novel emulsion solvent diffusion method, in which the solvent which is used, it is the ethanol dichloromethane solution it is used, and the polymer is anteric acrylic polymer is there, and it is poured, this polymer and the drug solution, it is poured into the aqueous solution of a PVA, which is the stabilizer, and that is thermally controlled at 40 degrees Celsius. The gas phase generated in dispersed polymer droplet by the evaporation of dichloromethane forms an internal cavity in a microspheres. The microballoon formed were floated continuously over the surface of an acidic dissolution medium that contains surfactant for more than 12 hours. You can see here in the diagram where the Udragate microspheres formulation, it has been shown. Udragate, it is insoluble, water insoluble polymer. So it will mix with the drug and then it is dissolved in an organic solvent. Then this solution, polymeric solution or polymeric organic solution, it is drop-wise, it is added into the aqueous solution that contains a stabilizer like PVA, polyvinyl alcohol. Then under this all procedure was proceed under magnetic stirring. Then after the solvent evaporation, it results in a formation of microspheres that is empty or having an internal cavity. Because of the internal cavity, its density is less. That's why we call it as a floating microspheres, which upon drying form the hollow microspheres. Then second form, it is known as alginate bead. The spherical bead of approximate 2.5 millimeter diameter can be prepared when the sodium alginate solution was dropped into the aqueous solution of calcium chloride, which leads to a formation of a porous system, which can maintain the floating for over 12 hours. Now, this system is little bit different, that it involves the formation of microspheres involve cross-linking process. Here, the sodium alginate contains carboxylate group, which is negative charge, that interact with the positive charge of a calcium ions and cross-link the microspheres which is formed. But such microspheres which form, it is hollow microspheres because it is porous in nature. and it will float for more than 12 hours. So who float for more than 12 hours, it showed a sustained drug delivery for more than 12 hours. So here you can see in the diagram the alginate solution. It was dropped into the calcium chloride solution. It resulted in formation of alginate bead or alginate microspheres with the evaporation of a solvent or evaporation of the internal solvent, which result in formation of a porous microspheres or microballoons that provide sustained drug delivery for over 12 hours. Thank you so much for listening me carefully. Thanks a lot.